Well, thank you for joining us for the third installment of the Science for the Community seminars. As I hope is obvious, I'm not Karis Mitchell-Moore. I'm standing in for her this evening. emceeing a little bit, introducing tonight's talk. For those of you at home in Zoom land, please, if you have any questions at any time, drop them into the chat. Then, I practice this. Ah, there we go. We can do that. Sure, try this. Okay, right. For those of you that are unfamiliar with the Chesapeake Biological Laboratory, it was founded in 1925 by Reginald Truitt. Those of you that are good with math, we're celebrating our 100th year anniversary next year. There'll be celebrations and opportunities for the public to join us in celebrating that. So keep your eyes peeled to the news. We'll be advertising it heavily, I'm sure. We're the oldest state-supported marine lab on the East Coast. There are 6 constituent labs that are part of what we call the University of Maryland Center for Environmental Science. UMCES is what we refer to it as. It's technically not part of the University of Maryland College Park. Many people think that that's the case. We are our own university. We're the smallest university in the university system of Maryland, but we are our own university. We are a world leader in the science of coastal environments, watersheds, and environmental education. Pick a topic in environmental aquatic science, and across the university, there's a researcher that specializes in that or is actively researching in that field. If you've been to any of the talks so far, you've probably noticed that there's a theme. Long-term monitoring and research, long-term data sets. Many scientists go out into any number of environments and collect single observations, but the ones that really have the power to guide researchers regarding how the environment is changing, how climate is changing, are these long-term data sets. And there are several researchers here at CBL that are collecting these long time series. And a few of them will be discussed this evening. Here's a good example of an ongoing collection with quite a bit of longevity. We've been collecting data at the end of the pier here, just right out front, for many, many years, since 1937, when the pier was constructed. And what we've measured has changed over the years: dissolved oxygen, chlorophyll, any number of things. But we've collected temperature consistently over that time. And that seems like a simple thing, temperature. But when you look at it over the long term and you compare the changes annually to that long-term average, it really paints a very telling picture about how water temperature is changing here in Solomons. The white would be sort of a neutral, blue would be a cooling year, and then the reds and the oranges are where we have a warming. The warming trend is pretty obvious. We always like to talk about dolphin watch. We have a program here called Dolphin Watch. We really like to bring this up at these when we interact with the public, because this is a citizen science effort. There's an app that you can download to your phone. If you spend any time on or around the water, you can just log on and log that sighting. And then that data goes into this huge database of sightings. We're in our 8th year now. Over 21,000 observations have been confirmed, 15,000 registered users. So we encourage you, if you spend any time on and around the water, please join us and become a citizen scientist. And then really at the core of what we do, in addition to great research, is we're training, we're educating, we're training the next generation of environmental scientists and environmental leaders. And we do that in the traditional format in the classroom by teaching, but also they get experience, hands-on experience in the field as they pursue their degrees, whether it be a master's degree or a doctorate in marine science or a related environmental field. This is the one plug this evening. If you feel like contributing to that effort, we encourage you to scan in or find us on the website. Any funds that you donate go to travel, enable students to travel to conferences, help fund the presentation of their work. So tonight's talk, we're going to have 3 of these young scientists that are the next environmental leaders that we were just talking about in the previous slide. Teresa Murphy, Nina Santos, and Lael Collins. They are involved in Dr. Woodland's laboratory and Dr. Wilberg's laboratory here at CBL. And they are going to talk about some of these long-term data sets, data that's been collected here right in front of the lab, and how that long-term data set is being extrapolated back to some of the newer science that's informative about what's happening with regard to our changing climate, our changing environment. And turn it over to Teresa first.
I apologize. I just got to get myself set up really quickly. Make sure I've got all of my I's dotted and T's crossed. All right. So thank you, Jerry, very much for that very generous introduction and a really good just introduction about why we're here, what we're doing, some of the long-term data sets that we're seeing. And now we get to talk a little bit more in depth about our little corner of long-term data sets. Which is essentially our Seines survey, which we'll be talking about in depth, as well as our benthic mysid survey as well. Okay. So to orient ourselves a little bit, it's kind of hard to see up top with the bar in the top there, but we are in a little place called Seines, Maryland. Which is located right in the middle of the Chesapeake Bay, which is actually the largest estuary in the United States and the 3rd largest in the world. And it is centrally located in the bay and representative of a lot of the habitats that are within the bay, especially since it falls like right in the middle of the salinity gradient that we have going on here. And it's widely applicable to a lot of shallow water nursery habitat for the fishes that we have in the Chesapeake Bay. And I mean, generally, the fish who thrive at the far end of each gradient are more likely to be able to tolerate and pass through this area, which is why it's so widely applicable. All right, so 25 years ago, our Seines survey started with a man in a dream. And that man in that dream is Dr. David Secor, fisheries ecologist extraordinaire here at CBL. And he focuses a lot on the community dynamics of fishes. And for a while, he was pondering this dream of how is the juvenile fish assemblage, or community rather, changing over time. And while dreaming about this, he was kicking around how he can do this. And in his mind, he was like, well, there's an extensive record of water quality monitoring that's being done right out in the bay, like Jerry had mentioned before. We're located right on the water. It's super easy to get to that habitat. And he has a lab full of students. So naturally, a Seines survey was born. And essentially what a Seines survey is, and now I'll talk a little bit more in depth for people who aren't familiar with the beach seine, is essentially, it's a big net, like the picture that you have in the lower right corner over there. It's a big net controlled by 2 poles, 2 operators, and it has a lead line on the bottom and a float line on the top so that it stays vertical in the water column as it gets dragged through. And essentially what we have been doing for the past 25 years is that we've been going out once a week, May through October, every week, we take 100-foot seine out into the low tide area right outside the pier here. And we do it at low tide because that's when it's most accessible for us. So the tidal, fluctuations change every week. So it really depends on what the tide is doing to what time we're out there. Sometimes we're out there at 6 a.m. Sometimes we're out there at 6 p.m. It really just depends. As long as it's in the light, then we're okay. And how we operate the seine, you can kind of see it in both of the pictures, bottom and up top, where we take the seine, one operator stays close to the shore, the other operator goes horizontally out into the water, creates a quarter arc sweeping motion around this way, comes back around to the original operator, and then we bring in the seine in. Hello? Okay. And then we drag in the seine, and it doesn't show it in that picture, but our seine actually has a bag where all the fish are collected. And this can either be done once on a survey day, or it can be done multiple times. It's kind of changed throughout the years. Currently, we're doing it once per week. And then once we get the fish on shore, we speciate the fish, we count the fish, we get lengths on them, and then we either, most of the time, we release the fish, or sometimes we actually harvest the fish depending on what projects are going on at the moment. If we need like muscle tissue or eye lenses or anything like that, we keep them. And I have some pictures up here just kind of showing what that process is looking like, us speciating them. The pictures to the right are some fish on our measuring boards. And that is generally what it looks like every Thursday throughout the spring and summer here at CBL. So I also compiled, again, still kind of hard to see with that bar up there, but I wanted to highlight a list of the top 15 species that we've been seeing over the years. And of course, although these species, they fluctuate very heavily in both number and size, but depending on the time of year, the week, everything. But you can really see that there's a lot of different fish that we're catching. I think in total, we've seen well over a few hundred species in total in the Seine. But these are just, again, the most numerous. So we see menhaden, striped bass, lots of silver sides, lots of anchovy. All of these guys we see pretty regularly. I also wanted to highlight some not so common but fun species that we actually do catch in the Seine as well. So we're catching pufferfish, we're catching burrfish, we're catching seahorses, and again, right outside of our pier, and also cow nose rays. And for a while before I came to CBL, and I'm sure some of you also did not realize that seahorses were actually here in the bay, but they actually are, and it's really exciting to see them coming back in better numbers since the SAB is increasing over time. All right, and now when we look at some of the data that's been coming out of the same survey, one paper I wanted to specifically highlight here is Wingate Seacore 2008, which has only used the data that is collected within the Seine and the water quality monitoring that is done off the pier, in which they looked at the effects of winter temperature and winter flow on the summer-fall nursery assemblage or humidity of a lot of the important fish we care about in the bay. And I know this is a lot to look at, so thankfully I have done a little bit of work, and just to sum it all up, I don't want to get too far into the weeds of this, but I did want to show the predictive power, the abundance and the incidence of these fish throughout time and season. And for example, the winter temperature and winter flow both have significant effects on these animals, particularly with the ones below each picture showing, they can either be lower in number in the following assemblage or higher in number. It really just depends. But again, this is just to show that this survey can really show the predictive power of what's happening with these animals in the following year. All right. So more on a little bit of that Seine survey data later. But for now, I also want to introduce, again, another man in another dream. And 2 years ago, my boss, Ryan Woodland, estuarine ecologist extraordinaire, and a former student of Dave's who we met earlier. And now Ryan loves these little shrimp-like crustacean called mysids, which are super important food sources for a lot of fishery species that we care about in the bay. They're chronically understudied, and he was generally dreaming about how are mysids using this shallow water habitat. And again, Nina and Lael are going to do a very extensive talk on what mysids are doing later on. But for now, that's what we're doing. And so with that in mind, a MICED survey was born. Now, here's a little overview of what we're doing with the MICED survey, much like the Seines survey. We're out there once a week throughout the year, and this is actually through the winter time as well. So generally speaking, we get about 51 to 50 data points every year through the survey. We take our custom sled, so you can actually see the sled that's over here on the table. It's like on its side, so it's kind of hard to understand based off of this, like how it's moving through the water column. But essentially, this sled has a net in between the metal casings of it. We pull it along the bottom and it's able to capture all of the animals that are located right at or just above the surface of the sandy sediment. So we go out there every week, take the sled, do some pulls. We have multiple transects on this, which we will talk about later. And we always do this at 11 a.m., not influenced by the tidal cycle, because MICEDs actually exhibit something called vertical diel migration, which we'll talk about later, but it basically just means that the MICEDs move up and down with the light every day. And you can actually see a bit of our transect over here to the left, because we did a little bit of MacGyvering, which was really fun, and we just strapped a camera on with some zip ties one day. And it's really nice to be able to see exactly what you're capturing through that method. All right, so now once we get the MICEDs on deck and we take them into the lab, what we do is we dump the sled out into a container, and the MICEDs are preserved either in ethanol or formalin. They are counted and speciated, and then we are used for our analysis, which we will talk about much later on. All right, I also want to talk about the professional and graduate impacts of these surveys. With the help from the Seines survey, research assistants and also postdocs have published works looking at menhaden and bay anchovy. And graduate students have actually used it in their dissertation theses to look at things like finfish nurseries and crustaceans, since we catch a lot of blue crab in the Seine as well, menhaden, community eDNA barcoding, and bluefish dynamics. And currently, just in our lab alone, we have 3 students using both of these surveys to complete their graduate studies with Angel, looking at weak fish from the Seine, Lael and Nina, of course, using, most like MICED survey data and Seine survey data. But again, we will touch much more on that later. And furthermore, past and present undergraduates have also benefited from these surveys by using them as opportunities for hands-on learning, and to support their projects like summer internships and undergraduate theses as well. We also have some great intra-collaboration happening within CBL, as it takes a village to have a survey going on like it has for the past 25 years. And we've actually had over 250 unique participants over those 25 years. And it's really great to see basically everybody from all corners of CBL come together and be able to bring this to reality. In terms of inter-collaboration, we also have a lot of collaboration with a lot of our local research institutions, including like the Calvert Marine Museum, who sometimes uses our collections if we find anything fun. They're able to take those guys and put them on display and use them for educational purposes. We also, as part of Morgan State University, work with Pearl in order to try to expand this MICED survey to look at other habitats as well. And I touched on it a little bit earlier, but we have undergraduate students from St. Mary's College of Maryland, College of Southern Maryland, as well as international undergraduate students who also work on these surveys as well through the Seas Coastal Alliance. And finally, perhaps one of my favorite things about the survey is actually exposure to the outside community, like you fine folks. Because I think it's really important to have engaging in science that's really visible to everybody and accessible to everybody. And since we're right in the middle of Solomons and everybody loves to walk by, enjoy the water, we're right next to the tiki bar, people like to walk around after that. It's just a really great thing to see the community engage and the community come out and ask questions and be able to interact with everybody as well. Now, since we're talking about a lot of the under the hood portion of these surveys, I also want to be really transparent and talk about monetarily speaking, in terms of like monetarily and time, what it takes to conduct these surveys every year. And between materials, time, and analysis, it's generally speaking about $1,000 per year on these surveys. Which, I mean, isn't a huge amount of money, but it is also isn't pennies either, but that's generally what we're kind of looking at there. And then, since we're also looking under the hood, I do want to share some of the struggles that we've had over the past 25 years. Because that's just the nature of keeping long-term surveys going, right? So, for example, sometimes we just have way too many fish. Menhaden, for example, fish that we see a lot, they actually can create these giant schools of fish. And some of you may have seen them like close to the pier, and like these big black balls of fish that are just roaming around. And they're constantly roaming around. And every once in a while, we will catch a giant big ball of fish, which, I mean, is fun, but it's also wild. So in cases like this, we do something called subsampling, which is still robust, and we're still getting the analysis and the data that we need, but less fish to actually count and, measure. We also go out, bad weather. Bad weather happens all of the time. You can see in the bottom photo, this is literally a day in January where the rain was going sideways. And it was very, very windy and very, very cold. But, as long as we're adhering to the safety guidelines that we're here, we go out. Also, sometimes things like in our epibenthic sled over here for the mysid survey, there's things like too much sand that happens sometimes. And, the environment is very variable, and that's okay. But that means that we have to get creative with our solutions sometimes. So tweaking our equipment is a must sometimes, and being creative is our friend. All right, so the main takeaways before I hand it over to Nina to talk more about the data portions of these surveys is that I want you to understand these surveys and how they shape what we know about long-term ecological trends. I want you to also understand the under the hood mechanics of these long-term surveys, and also see how far these types of surveys can reach the impact. Right, and we'll take away Nina.
Cool. Okay, so as Teresa sort of introduced, we have our Seines survey and our mysid survey. In our Seines survey, we're capturing fish. I assume you all know what a fish is, but you might not know exactly what a mysid is exactly. So this is a video of some mysids that we had in the lab that are sort of swimming around. They're small shrimp-like crustaceans. They're not true shrimp, although they're called mysid shrimp. They're also known as possum shrimp. I don't know if you saw in an earlier photo, one of these guys that Teresa was showing had sort of like a pouch. So the females of female mysids will have a pouch when they're sort of carrying their young, and then they'll release them into the water column when they've developed a bit. And they're a type of mesozooplankton, as Teresa mentioned. They're chronically understudied, like Teresa mentioned. They can be really, really hard to sample effectively. I know in the past when I first started here at CBL, Brian and I went out a bunch of times using just like a normal like zooplankton net and pulling it across the surface and hoping to catch mysids. And then, later on we sort of decided that the sled might be a better tool for that, and it ended up being a lot better. So we sort of figured it out here, but it took some trial and error. They're also really hard to identify in stomach contents, which Leah will touch on a little bit later. And like Teresa mentioned, they're really important prey for a lot of economically relevant species, such as summer flounder, Atlantic croaker, striped bass, and weakfish, among others. And so this figure here is just showing some predators of mysids. So these colors here are just showing you different species like croaker, sea robin, scup, hake, striped bass, and things. And then the figure is showing the y-axis is just the percent of mysids that were found in the stomach contents, and the x-axis is just those different fish species. So you can see that depending on the species, there are a lot of mysids being found in their stomach contents. And up until, our lab trying to really understand more about mysids, like I said, they're really chronically understudied and we don't know a lot about them, but they're clearly very important for some species. And as Teresa mentioned, mysids are also really interesting because, like a lot of other zooplankton, they undergo diel vertical migration, which is just as Teresa sort of alluded to, it's a daily pattern of movement by a lot of these aquatic organisms between the surface and deeper layers of the water. And basically they're sort of hanging out on the bottom or in deeper parts of the water during the day, and then coming up to the surface at night. And then once the sun starts to come out, they'll go back down. And additionally, I wanted to introduce you guys to the idea of a food web. So we all know what a food chain is. You know, you got your phytoplankton, for example, and that's being consumed by a zooplankton, and that's being consumed by a small fish, and that's being consumed by a bigger fish or a shark. But in reality, we don't really see a food chain a lot in nature. Usually it's a bit more complicated and things are connected in a lot of different ways, like this sort of figure is showing, where, you know, not everyone is just eating one thing, as a food chain might suggest. Things are a lot more complex. And so where do mysids fit into the food web? So they're very flexible omnivores, which just means that they're eating a lot of different things. They're eating all different kinds of phytoplankton. They're eating different sizes of zooplankton. They're eating, you know, detrital material, just like some gunk that's floating around. And they also play a really important ecological role in coastal food webs called benthic pelagic coupling. So like I mentioned, mysids are sort of hanging out at the bottom during the day, coming up to the surface at night. And so they're being consumed by both pelagic predators, so fish that are hanging out in the surface waters, as well as benthic and demersal predators that are sort of closer to the bottom. So they're acting as sort of a connector for those benthic and pelagic food webs. And so why do we care about coupling food webs? Well, it helps to provide resilience to aquatic ecosystems because mysids are able to respond to changes in the productivity in those food webs and basically act accordingly. And so if there's more of a food source or less of a food source, they're able to switch their sort of prey preferences and what they're consuming to match that change. And so what are we doing in our lab to further understand mysid's role in Chesapeake Bay food webs? So the first is the mysid survey. And so this photo here is our CBL Pier, and it's sort of, you know, from the street there and then going outward into the water. You can see all the little black dots are the oyster cagers that are out there. And so the green circle is where we do our shallow sled pulls. And so we have two that are sort of happening in that green circle. One of them happens, you see my cursor, yeah. So one of them happens sort of along the pier, and we call that our shallow structured habitat. So that area has a lot of structure because it's along the pier. And so a lot of animals prefer that kind of habitat to others. And we weren't sure which mysids might prefer. So that's sort of something that I'll talk about a little bit later. But our second sort of transect that we do here is sort of also in this green circle close to the start of the pier, but we do sort of a diagonal, my cursor. So a diagonal sort of transect where we're going from the pier sort of to the shoreline. So we're getting sort of, instead of going along the pier with all that structure, we're going over that sandy bottom. And then the final transect that we do is in this blue circle, where we're doing sort of a deeper part of the water by the pier off into the floating pier. So now jumping into some of the data from this. So this is our monthly mysid counts from 2023. The y-axis is the number of total mysids that were caught in that month, and the y-axis is the month, or x-axis is the month. And so our colors here, the blue color is our shallow open transect, and the yellow is our shallow structured habitat. And so we can see that there's definitely like a seasonal difference in how many mysids we're catching. So you can see that in January through June, there's not a ton of mysids, but then in July, we see a huge boom in the number of mysids that we're able to collect. And we can also see a difference in how mysids are using these different habitats. So you can see in that yellow color, which is the structured habitat, it really seems like we're able to capture more mysids. And so it seems like mysids are preferring that structure. And now this is that same figure, but now I've added those dark blue bars, which is our deep habitat transect. And so you can see that typically there are sort of mysids in this deeper habitat almost all year round in comparison to sort of the seasonal trend that we were seeing for the shallow habitat. And so Teresa mentioned earlier that something that we do with our mysids is that we're speciating them. And so you might be wondering how we're able to tell different mysid species apart. So this is, the top picture is sort of a mysid from a side view, and then the bottom picture is from above. And basically, that little part of the mysid at the very middle of their tail fan is called the telson, and that's pretty much, for a lot of mysid species, all you really need to be able to tell the species apart. So this here, A through G, are just different shapes that telson can have. And that tail shape is all, all of those are different species. And so if we just look at some of these mysids under a scope, we can look at just that telson and the shape of that telson, and we can tell what type of mysid it is. And so that brings us to some work that was done by another student of Ryan's named Danny. She was a master's student that finished up a bit ago. And in her work, she was looking at sort of mysids, not only at the CBL Pier, but she was looking at mysids sort of along the Patuxent River. And so for her work, she went out in multiple months in, I think, 2018, and she looked at sort of here in the green is the Neomysis mysid species, and then the purple is Americomyces, and we can see that in May we have a lot of sort of mysids at P1, which is just one of the stations in the Patuxent River, and not a lot elsewhere. And at that point in the year, there were none of that purple species. And then in June, you can see that she caught more at the second site and a bit more at station 4. And still in June, there weren't a ton of that other species. But then in August and September, it seems like we sort of have a shift where both species are pretty abundant in August, but then in September, that green species, or the Neomysis, really declines. And it seems like the Americomyces is taking over. So not only through these surveys and through this work are we able to see sort of these seasonal dynamics, we're also able to see that different species are having different dynamics and different relationships with the seasons. And so a major thing sort of in the Woodland lab that we do is studying food webs. And so how exactly do we do that? So one of the major ways that people study food webs is by looking, you catch a fish and you look at what that fish is eating, you cut it open, look at its stomach. And that's called stomach content analysis. And this is great. And Lael will touch more on this later with her work because she does some stomach content analysis. And this is great because you can look into the stomach of the fish and you can say, okay, that fish ate 3 mysids, it ate a bay anchovy, and it ate a worm. And so it's great species level data. The technique has been used for a really long time. And it gives us a really great snapshot of what has most recently been eaten. However, the technique has its challenges. So sometimes you'll open up a stomach and it looks like that. And I don't know about you, but I don't know what any of that is. So it's really hard to say, this fish ate a bunch of gunk. So that doesn't tell us anything about the food web or how that fish is related to the rest of the organisms in your ecosystem. So that's where another part of sort of how we look at food webs comes in, and that's called stable isotope analysis. And so what are isotopes and how do we use them? So the entire premise of using stable isotopes is that you are what you eat plus some biochemistry. So it's a powerful tool that basically is hinged on the idea that your diet, whatever you're eating, is imparting a chemical fingerprint onto your tissues, and it can give us a more integrated view of food web dynamics. So what do I mean by that? So like I mentioned before, the stomach content analysis, you can cut open a fish, you can see what it ate, in the last 12 to 24 hours. But, if you cut me open right now, I ate some chicken nuggets, but I don't eat chicken nuggets every day. So using something like stable isotopes, we're able to get a more integrated view over weeks to months of time, rather than just a quick snapshot of what was eaten that day or the day prior. So in case you can't remember back to high school chemistry, isotopes are atoms of the same element that have the same number of protons, which means they have the same chemistry. a nitrogen atom is a nitrogen atom. But they have different numbers of neutrons, which means that they are not the same mass and they have different weights. So for example, in this picture, I have sort of a scale with a nitrogen 14 and a nitrogen 15. They both have 7 protons, but the nitrogen 14 has 14 neutrons, the nitrogen 15 has 15 neutrons, and so they weigh differently. And that, you know, that one neutron, it might not seem like it has a lot of difference between these two atoms, but in these biochemical processes that we're talking about, and you know, an organism consuming food, having these biochemical processes, excreting, you know, waste, that weight makes a difference. And so we use these isotope ratios, looking at the relative number of these heavier to the light isotopes present in a tissue sample, for example. And they're really useful for looking at food web dynamics because these isotope ratios change in predictable ways as organic matter moves through a food web. And they can help us to identify patterns of trophic dynamics and tell us where an organism is sort of in a food web. And what do I mean by that? So typically in stable isotopes and in ecology, we're looking at carbon and nitrogen, which are really abundant in sort of organisms, tissues. And so typically the way that you would look at this data set or this type of data is you would have the nitrogen value on the y-axis, you'd have the carbon value on the x-axis. And this is a very simple food chain that I'm showing here, where we have a phytoplankton at the bottom, and then we move up a little bit to a zooplankton, and then to a small fish, and then to a large fish. And because isotopes, as we're moving up the food chain, they change by a predictable amount, that ratio of heavy to light isotopes, we can predict, you know, if we caught that top fish as a codfish, if I were to catch that codfish, run its stable isotope value, I could say, okay, well, there's been 3 trophic transfers on average that this fish has experienced, which means that it is a trophic level 3 or 4 or whatever it ends up being. And so some takeaways from this part of the talk, a lot of this work is ongoing. So as you can see, I didn't show any real isotope data today. That's sort of part of my dissertation that's ongoing. Maybe someday I'll be back to talk more about that. But I hope that you can take away that mysids are really important to Chesapeake Bay food webs, but also in aquatic environments all over the world. And that our lab is really wanting to understand food web architecture because it's important for predicting how food web linkages might change, and how we can buffer that change in the face of a changing environment. And now I'm going to turn it over to Lael, who will talk more about how she's using both the mysid and Seines surveys together.
Yes, thank you, Nina, for that sort of introduction. So as Nina just mentioned, I'll be talking about a big chunk of my master's project where I'm actually using data from both the Seines survey and the mysid survey, and kind of looking at interactions between fish from the Seine and their impacts on mysid communities. So I won't go super in depth about mysids because Nina did such a great job, but just to kind of reiterate, mysids are typically seen as important prey items for lots of organisms in Chesapeake Bay, and they are also pretty mysterious in the sense of they are pretty understudied, and we don't know a lot about the specific types of relationships that they have with other organisms. It's because of this that for my project, we look at the impacts of predation on mysid communities to understand the impacts that fish are having on these pretty important creatures. So as it's been said already, I do use data from both surveys that we've talked about. I just want to reiterate for this slide that we do multiple tows for each mysid survey sampling day, where we have 2 tows that are in the shallow end and then a singular tow in the deep end. When I'm using this data, I'm ultimately using it to come up with abundance estimates. And so for those kind of purposes, you can look at the data, kind of like as is at the different toes, but also you can aggregate it and come up with kind of like single values for each sampling day. Teresa's already talked about kind of like our processing steps once we have this data. And again, I just wanted to kind of bring in the fact that it is used, you can kind of use it for like multiple things. And for a lot of my project, I use like aggregated values. And so I use averages of the different toes from each sampling day. When I show data later on in my presentation, that's kind of what the data that we're working with is. So we've also talked about the Seines survey, and that is where fish from my project comes from. And when we're seining, we seine in an area that's very adjacent to the pier. So the habitats overlap a great deal. They're very representative of one another. But here's just an aerial view of kind of like the Seine area specifically, where I collect fish for my specific project. We've kind of talked about what some of the things that you can do with the fish, like Teresa mentioned. There's islands analyses. You could do a stable isotopes analysis on the fish as well. For my purposes, I dissect their stomachs and I conduct stomach contents analysis. So for that processing, they're frozen until it's time to dissect them. When it is time to dissect, I take length and weight measurements, as well as the full and empty stomach weight values. From there, I empty out the stomachs and I actually look at them under a microscope to just visually observe what I'm seeing inside of these stomachs. And Nina kind of touched on this, but down kind of towards the bottom left or bottom right, I have two different stomach contents. One is from a spot, and that's this one that's more towards the middle of the screen. And from that one, you can tell that there's not a lot of sense to be made with what's in there. This is what, unfortunately, a lot of these stomach contents look like. You can see that there is material, you just don't necessarily know what it is. More towards the right is the stomach of a spotted sea trout, which was a very nice stomach to analyze. You can see that there are some mysids in there, actually. The mysids are the paler sorts of organisms. So here's one, here's another one with the like bigger black eyes. And they're making up a lot of this stomach, but there's also other things like amphipods, which are just other benthic, small like crustaceans. And then there is a little bit of unidentifiable material, but not to the same degree as what that spot stomach is giving. So this is just kind of a range of what some of that data that I'm getting from the fish looks like. So I'm going to talk a bit about the specifics of my project now. My master's project kind of stemmed from work I did as a research assistant for about a year before officially becoming a master's student. And in this year, we did kind of like a similar project. I was quantifying the consumption of mysids by finfish. But this project came from the 2022 Seines survey. And so all of this data was collected during our daytime seines. And ultimately from this, there wasn't, the predation on mysids wasn't necessarily as high as we would have probably expected. We found very low predation rates. And there were very few species of fish that we were catching that are likely to be consuming mysids. Usually in the literature, the kind of more voracious predators of mysids are the fish that fall into the drum or maronid families. What's interesting though, is that also in 2022, we did a singular nighttime seine. So this wasn't a whole entire survey. This was just one instance that we sort of did out of curiosity. But what's really interesting is that from this nighttime seine, we found a great deal more evidence of predation on mysids. And the fish that we were catching are ones that we would typically expect to be eating more mysids. So going into my master's project, we focused on more specifically on nighttime seine sampling, which is kind of like the addition of another seine survey in addition to the yearly one that we have going on. So, like I mentioned, going into 2023 and my master's work, we are able to kind of compare data that we got from 2022, where we had very low predation and very low consumption values, and compare that to things to data from 2023. Before I kind of get into those specific comparisons, I just want to go over some mysid data, which of course was made possible due to our mysid survey. And pretty much in these figures here, specifically the figure on the left, this just shows generally the estimated number of mysids we would expect to be in our survey area between the 2 years. And this is just an average of all the tows for the time period that I'm looking at, which is August to September of the 2 years. You can see in this figure that there's not that 2023 looks like it has a few more, or a bit more of a mysid estimate, but these values are not so different that we would be able to say that any differences in the data are due to there being a great deal more mysids from 1 year to the next. Now, I mentioned that I'm using data specifically from August and September for the 2 years. And the figure to the right, I just have the aggregated mysid estimates from 2023, just to show kind of mysid, our estimated abundances from that time period. From just generally from this, you can see that there are fluctuations in the numbers, and that is something that I think would be really cool to look into. I just have not done that yet. Here I have results from our 2 surveys that we're looking at. So just to remind you, I have daytime data from 2022. This was when we were finding there to be less predation on mysids. And I've also incorporated nighttime data from 2023. And what's just kind of interesting from this data is that, like I mentioned previously, the types of fish you would expect to be consuming mysids are usually going to be your drum and maronid families. What's particularly interesting is that you can see here that I've highlighted the drums and maronids. They're typically more abundant in from our nighttime survey that we're looking at. So that's just something kind of right off the bat that's pretty interesting to take notice of as I'm talking about this data. Also from the same survey, I have here the species of fish that when I dissected them, I found positive identifications of mysids in their stomach. And so this plot just shows an estimate of, based off of the counts of the species of fish from the seines that we did, the numbers that we would estimate to be in the study area as a whole for that August-September time period that I mentioned. So the species are Atlantic croaker, Atlantic needlefish, silver perch, spot, and white perch. And just from this, you can see that Atlantic needlefish are kind of dominating those other species. There are a lot of those out at night from the nighttime survey. And the other ones you can see, they're there, but not nearly to the same degree as how we were catching the needlefish. What's interesting about that, though, is that on this plot, I have those same species of fish along the x-axis, and I actually have the average numbers of mysids in each of the stomachs, as a species, that I counted while dissecting them. And so this one's particularly interesting because while needlefish were dominating in terms of the pure abundance of the fish species, the white perch are pretty much dominating in terms of consuming mysids. Croaker and silver perch are also eating a decent numbers of mysids, but not to the same degree. And then needlefish and spot were, again, very few mysids found in their stomachs. So I was able to use that data together, the abundance estimates of the fish, as well as the consumption by each species, to come up with consumption estimates of mysids by each of these species of fish. So I have, again, the species along the x-axis and the number of individual mysids that we would expect to be consumed based on the estimates of each of these species of fish. And what you can see from this is that the croaker, needlefish, and spot all had pretty negligible amounts of mysids that were being eaten, but the silver perch and white perch were consuming many more mysids, especially those white perch. They had the highest consumption value. I also went and calculated the percentages of the mysid populations that were being consumed by each of these species. I did this again using those consumption values over the number of mysids that we would estimate to be in the areas, to see the proportion of mysids that were being consumed. So this follows that sort of same pattern, where silver perch and white perch are consuming a good deal more of fish. So the main takeaways from this kind of comparison are that the fish community did have a difference in assemblage between day and night, and dominant predators of mysids were really only observed from that nighttime survey. And this was likely the main reason that, or a large reason for why we calculated such much higher predation rates from our nighttime survey compared to the very low values from the daytime survey the year before.
All right. Now, really quickly, I know we're running a little short on time, but the three of us did want to talk and kind of wrap it up, and kind of talk about the final conclusions about everything that we've been talking about, kind of send you home with us, like a take-home message. And so generally, why do we need long-term survey data? What's the purpose? What are we really trying to get at the end of the day? Well, generally speaking, at the end of the day, I'm sure I don't need to tell all you lovely people that the world is changing. The world is changing in terms of land development. I mean, being a Southern Maryland native all my life, the place that I live in certainly does not look the way that it currently does now, even over my 29 years of being on this planet. So the planet, is changing in terms of land development, is changing in terms of climate change. And we really need a way in order to quantify what we're seeing and where we're going, generally speaking. And I mean, in terms of long-term data sets with the Seines survey, for example, like the 25 years that we've had it, we've already seen some pretty major changes that we've talked about earlier, and in the fish assemblage that we're seeing just here right outside of our pier. And in order to get more accurate data points like that, we need to have long-term data.
Yeah, so another great benefit of having these long-term data sets is that you kind of like what I just talked about with my specific project. You can kind of almost take snapshots in history. You can take like a very specific time period and focus on that. And even when you're taking smaller portions from that, you can kind of contribute to like grander ideas, to the point where you even make a master's project out of it, maybe even PhD, or at least kind of build up, these sorts of projects. And so, that's just kind of like another benefit on, from a different perspective of these, surveys.
And then finally, we just really wanted to highlight the fact, this really long-term survey of the Seines survey going on for 25 years, and the MySID survey is relatively young, but we're planning on doing it for the foreseeable future. And just, thinking forward, and hoping that we can do more work like layouts where we're sort of coupling these two long-term surveys and able to look more at how species are interacting and looking at how food webs are changing as sort of the changes that Teresa mentioned are happening. So.
Without further ado, thank you. Thank you for everybody who is here.